Hello, welcome everybody to this fifth session of the SICAM webinar series, The Importance of Being H.P.C. Earnest. This is the last one of this series of meetings motivated by the impact that H.P.C. has on simulation and modeling. In this series, during these sessions, we have been inviting leaders of collective initiatives, projects, institutes in Europe and USA, which focus on the challenges of the development of scientific codes, algorithms, and data-driven initiatives in the context of the future, the coming H.P.C. landscape. We have been organizing them according to the SICAM spirit, and with the idea to bring together complementary initiatives that focus on the science that is made possible by combining modeling methods with optimal exploitation of H.P.C. resources. We have always aimed to fostering an open and informal dialogue, which is also interdisciplinary. And to that end, we have always been inviting representatives of these initiatives that explore complementary areas within the broad SICAM community. And in this last session, it is a pleasure to introduce our three speakers. So we have Stephen Louis, who is professor at the University of California at Berkeley and also senior scientist at Lawrence Berkeley National Lab. He is well known for his groundbreaking work on the ab initio GW method, and also because of his contributions in the area of low dimensional systems and in nanostructures. He's today with us also because currently he is the director of the C2SEP-EM. This is the Center of Computational Study of Excited State Phenomena in Energy Materials. And the C2SEP-EM is one of the DOE-funded centers in the States. And this institute in particular, has the mission of developing new concepts, methods, and software for excited phenomena using ab initio quantum many-body systems. I'm sure we will hear about this today and about the science that is being developed in this context. Our second speaker is Claudia Draxel, who is Einstein professor at the Humboldt University in Berlin, and it's also a Max Planck fellow at the Max Planck Graduate Center for Quantum Materials. Claudia is well known for her leading research in the developments of theoretical concepts and the methodology of ab initio computational material science. And in particular, she is developer of the all-electron full potential package exciting. Claudia, in this more recently, is also one of the leading experts in the use of big data in material science. And she's the founder of the NOMAD Lab. Actually, NOMAD is one of the centers of excellence funded by the European Union. And she's also founder of the FAIR DI Association, which is a data infrastructure to enable data sharing and collaboration in data-driven science. I'm sure we will hear about challenges and opportunities in data-driven initiatives today. And our last invited speaker is Modesto Orozco, who is director of the Integrative Biology Program at the Institute of Research in Biomedicine in Barcelona. Modesto is recognized by his outstanding work in the understanding of biological systems from first principles. And specifically, he has leading contributions on the understanding of the mechanisms of signal transduction in proteins and more specifically in the chromatin. Modesto has a leading role in a number of computationally driven initiatives in biophysics and bioinformatics. In particular, currently he's one of the team leaders in BioExcel, which is another center of excellence that we heard about earlier in this series. And he's also involved in Elixir, which is an intergovernmental organization that brings together life science resources across Europe with the idea, these resources include databases, software tools, training materials, and the goal is to coordinate them to have an agile infrastructure that facilitates sharing of expertise and also to identify best practices in the communities in bioinformatics and computational bioscience. So I'm sure we will have three very engaging lectures today. We will stick to the usual format. We will first listen to three presentations, one after the other, and we will have a common discussion and question and answer session at the end. This will be chaired by Sarah Bonella, the CICAM Deputy Director, here with me and myself. We will take care of managing this discussion session. You can ask your questions using the YouTube channel that you have, or otherwise you can also send us your questions by email to the email address director@cicam.org, and we will collect all of them and distribute the questions at the end of today's session. So without further comments, I think we can now move on, and I give the word to the first speaker of today, Stephen Louis. So Stephen, the floor is yours.
Okay, thank you. Okay, thank you very much, Ignacio, for the very kind introduction. In this talk, I would like to share with you some of our work in our center to study quantum interaction phenomena in materials using H.P.Cs. And this is the outline of the talk. Since this is a very broad audience, I will begin with some very basics, and then we're going to discuss the ab initio study of many electron interactions and excited state phenomena in materials. And then toward the end, I'm going to discuss some present status and computational challenges regarding to these areas of research in the condensed matter and materials physics. So as you probably all know, the properties of material are basically dictated by the interaction of the electrons and ions in the system. So this is a known problem. The difficulty for the problem, solving this problem, is that you have enormous number of particles strongly interacting in your system of interest. For example, if we look at a coin, say a penny, a US penny, then there are actually as many electrons in a penny as there are stars in the known universe. It is this strong interaction between many particles that give rise to many of the emerging important and interesting property of materials. And this is also one of the reasons for the richness of the field of materials physics. So straightforwardly, one could say that if I solve the Manybody Schrödinger equation for your material interest, then you should be able to obtain everything. And of course, this is the non-relativistic form or the Manybody Schrödinger equation. Unfortunately, for a system of interest, typically a material would have 10 to the 22nd particles per cubic centimeter. Of course, it's impossible to solve exactly such a system, quantum system with so many particles. In fact, it's often not desirable to obtain the full solution for the problem because the exact wave function would be so complicated involving 10 to the 22 variables. In fact, if we were to try to understand the property of a material, it's very often better to look at quantities that are more closely related to experimental measurement. For example, if we are interested in understanding the transport property, say the electrical property of a material, then it is more appropriate, in fact, to look at the probability of finding an electron, injected electron in a system at position r and some time tau later after you injected the particle in position R prime and at time 0. So this, in fact, is exactly related to a transport measurement. And this expression, this probability amplitude, in fact, is mathematically the interacting single particle Green's function of an interacting many-body system. So if we could solve this problem, then we could obtain the optical, the transport property of material. So this is what we call an n+1 particle problem, in which you try to focus on the behavior of one excited particle in a system of n particles interacting. So why are excited state phenomena important? They are really important because phenomena such as transport, optical, energy transfer, charge and spin dynamics in a material often give rise to its defining attributes, whether it's a semiconductor, superconductor, photocatalysis, catalyst, and so on. And it determines its usefulness. There are actually now many existing ab initio methods and codes for studying excited state properties in materials. They are very successful in predicting and understanding 1 and 2 particle excitations for increasingly complex materials. Here I list some of the common community codes that are available now. However, there are also many key phenomena, such as free-of-Hall particle correlated excitations, nonlinear spectroscopies, field-driven time-dependent processes, and excited state dynamics, and so on, had yet to be addressed adequately by ab initio methods and public domain softwares, which would include all the relevant and accurate many-body, many-electron interactions. So what I want to do today is focus on looking at the electronic properties, excited state electronic properties in materials. So because of this interacting many-electron problem, we need to look at this problem from the point of view of using many-body techniques. And in fact, for moderately correlated electron system, one could numerically obtain the solutions for the property of interest using, from first principle, using many-body perturbation theory. For example, say the GW method or the GW beta subunit equation method. In the GW method, which I will discuss a bit later, one is focused on 1 particle excitations. So this is the n+1 particle problem I mentioned. Here you solve the 1 particle Green's function. And then if you're interested in the optical properties, then you're creating an electron on hole in the optical process. And this is an n+2 particle problem. And you have to look at the 2 particle Green's function. Now, let's first look at quasi-particle excitations in solids that involve looking at the 1 particle Green's function. Within many-body theory, the dispersion relation of your excited particle can be written in terms of a term that corresponds to the non-interacting system, some kind of mean field, plus a self-energy that will modify the dispersion due to interactions. And this self-energy operator can be expanded in terms of, in perturbation theory, in terms of the interaction. Normally in textbooks, such an expansion will be in terms of the Coulomb interaction. Within the GW approach, what you do is you reformulate the problem so that you expand the perturbation theory in terms of the screen Coulomb interaction, where here is the dielectric screening of the material, the particular material of interest. Now, because the screen interaction is much weaker, so this series, in fact, is expected to converge very rapidly. And the GW approximation involves just taking the first turn in this expansion. And that turned out to work quite well. Then you're interested in optical, linear optical properties, then we have to solve the two-particle Green's function. And then you have to include effectively two-particle interactions. At the end, in both type of studies, what one end up with is a very large, dense, non-Hermitian matrix eigenvalue problem. And the dimension of such a matrix can be as large as 10 to the 7th or even larger. I will give you some example later. However, this kind of approach, in fact, works quite well. If you look at the quasi-particle excitation, for example, the band gap of insulating materials, here is a plot of comparing the theoretical band gap compared to experimental measured band gap of semiconductor insulators. If there are perfect agreement between theory and experiment, then the data points will be along the diagonal. So you see that if you were to interpret the Kohn-Sham eigenvalues from, say, a DFT calculation, in general, they would be significantly off compared to experiment. But the GW approach, in fact, works very, very well. And on the right is song is the optical absorption spectrum, which is related to the imaginary part of the dielectric function of a material for silicon. So here is the blue line is one when you neglect the electron-hole interaction. And then the red line is when you solve the two-particle Green's function to obtain the absorption. And you see that including electron-hole interaction, in fact, significantly changes the spectrum and may bring it to much better agreement with experiment. This kind of many-body effects, many electron effect on the excited states of material, in fact, becomes even more dominant in reduced dimensional systems or in heterogeneous systems. So here, let me show you one example of at the interface between inorganic and organic materials. That is, for example, I'm looking at a molecule on top of a metal. So how the molecular level aligns with the Fermi level of a metal will determine much of the property of such an interface. As we all know, if you bring a molecule from isolation to next to a metal, you would induce changes in the molecular level. We call that the level alignment problem. And there are several physical effects. One is interfacial charge transfer. Another one is quantum mechanical coupling that involve both one electron and many electron effects. And then there's also an electron self-correlation or self-energy effects that are going to come in. So let's look at the example of a benzene molecule on top of graphite, which is metallic. If I were to do a LDA DFT calculation, LDA stands for local density approximation within the density functional theory, then what you find is the HOMO-LUMO gap of the molecule is at 5.1 electron volt when it's in isolation and vacuum. When it's right next to the metal, you keep it at a van der Waals distance away, it doesn't change much at all. It stays around 5.1 electron volt. If you do a GW calculation, what you find is that that HOMO-LUMO gap on graphite, in fact, is 7.3 electron volt, significantly different from the Kohn-Sham gap. And finally, if you look at an isolated molecule, then the HOMO-LUMO gap, in fact, is 10.5 electron volts. So there are significant many-body effects that we normalize the HOMO-LUMO gap as you bring the molecule toward the metal. And within DFT, what you find is the Kohn-Sham gap would stay the same everywhere because it couldn't see the metal if it's at a van der Waals distance away. And in general, this kind of self-energy effect is very large, for this case in the order of 2 electron volts. So let's consider the same molecule now on different type of surfaces. If I were to do a LDA calculation, then this is the gap for the different surfaces. If you were to do a hybrid calculation, then again, there's the gap doesn't change whether it's in vacuum or in different on different surfaces. However, if you do the GW calculation, then you see there are significant difference for the same molecule, benzene, on different surfaces. That is basically due to this self-energy effect. And in fact, depending on which surface that you put the molecule on barium oxide, the band gap could be very different because there are surface states on this particular surface. So this is just to illustrate that the strong and different renormalization of molecular level with different contacts are absent in the kind of Kohn-Sham orbitals mean field theory, even if you do hybrid calculations. Now, this kind of effect is also becomes very dramatic also when you look at lower dimensional systems, such as 2D materials, which are of great interest lately. Suppose I put one of this system, 2D system, on top of a substrate. This is a case of a monolayer molybdenum on top of bilayer graphene. Here I illustrate the effect of screening or interaction between the substrate and the system of interest. If you put an electron here, in fact, it will induce, excuse me, I think I'm going the wrong direction, induce a screening charge, a non-local screening charge. And this screening effect is non-local frequency dependent, and one has to fully account for this in order to understand the property of this material in such an environment. What we find for some system, in fact, for some 2D systems, substrate and encapsulation can renormalize the band gap by bigger than 50%, and changes the exciton binding energy by a factor of 2 or more for such systems. And one could also use the different substrate to tune the energy of the gap and even the energy of plasma in such a system for various studies, science studies and applications. Now let me turn to the software that we use to do this calculation. There, as I mentioned, there are many in the community now. I'm going to focus on the package that we developed, the Berkeley GW package. So it is a general excited state phenomena code that supports 3, 2, 1, and molecular system for doing GW and GW beta saltpeter equation calculation. It supports insulated metallic and semi-metallic systems. It interfaces with a large number of DFT and other mean field codes so that you could carry out this many-body perturbation calculation. It's massively parallelized. In fact, it could linearly scale up to about 500,000 CPUs at NERSC Core 2 machines. So this illustrates the speed up with the number of cores, and it can efficiently sample K-points for experts in the audience up to about 1 million K-points in the Brillouin zone. So these are very important for some spectroscopic studies. Example of some recent very large system we have studied is to look at the quasi-particle and the optical excitation of defects in silicon and silicon carbide. These are defects that has importance in quantum information studies. We have studied up to about 3,000 atoms or 1,100 electrons in a supercell. I'll come back to an example of that later. So the challenges in this field is that ab initio study of excited state actually require different and higher level treatment of many electron interaction, say from that of say DFT or other mean field theories. The need now is to able to develop more powerful software for increasingly complex materials so that we could look at 1 and 2 particle excitations. However, we're also in need of developing methods and public domain software to look at other key phenomena that has been underexplored in the past. I mentioned some of that already, so that one could include detail many electron effects in those phenomena, and one would hope to able to study this phenomena in detail using future exascale computing. So this led us to the establishment of the center that Ignacio mentioned earlier. To meet this set of challenges, we need a team of physical scientists, applied mathematicians, and computational scientists. So now let me tell you a bit about our center. This is a Center for Computational Study of Excited State Phenomena in Energy Material. We call it C2CEPN. It is a DOE-supported computational material sciences center. There are 5 of those centers now in existence in the US. It was established about 4 years ago, a little bit less than 4 years ago. Our mission is to develop and implement new theories, methods, algorithms, and computer codes to explain and predict excited state phenomena. There are 11 co-PIs from 4 different institutions forming this center. I'm the lead PI, and the center involves physicists, chemists, applied mathematicians, computational scientists, and a couple of experimentalists to keep us honest. So the main goal of the center is to go beyond GW and GW beta salpeter in the traditional sense. We would like to able to study from ab initio high number particle excitations. For example, Byexiton, which are correlated 4 particle excitations, trions, exciton-exciton interaction, decay of excited states, charge separation, exciton-phonon coupling, and so on. To study this set of phenomena, one needs to solve the interacting 3 and 4 particle Green's functions. Also, we would like to look at time-dependent and high-field phenomena. For example, pump probe experiments involving excitation of the electronic system, nonlinear optics, field-driven phase transformation, and so on. And there, we need to look at the Green's function explicitly in terms of time propagations. So we have going to we will have to look at time-dependent GW using the non-equilibrium Green's function formalism. So let me say a few things first about correlated multi-particle excitations. These are phenomena that actually very prominent in atomically thin 1 and 2D systems, such as the transition metal dichalcogenide 2D systems. For example, here I saw an experiment where what's plotted is the photoluminescence as a function of photon energy for different intensity of excitation light. And there's a one peak, in fact, grows with excitation intensity. And this has been identified as a 4-particle Byexiton excitations. And so this kind of excitation actually dominates the spectroscopy of 2D materials and, in certain regimes. And they are also important in understanding other fundamental and practical processes. For example, if one would like to look at exciton condensation or excitonic insulators, then we would like to know what the exciton-exciton interaction is like. And also for the case of singlet fission, that is one exciton in the singlet state divided into 2 excitons will be related to this kind of processes. So these are very important in terms of looking at improvement in photovoltaic inorganic crystals. So just to illustrate the complexity of looking at 3 or 4 particle correlated excitations. Here I illustrated by showing you the number of Feynman diagrams will be involved. In looking at quasi-particle excitations, let's say like the band gap of a semiconductor, we need to solve 1 Feynman diagram. For the linear optical properties, we need to solve 2 Feynman diagrams involving the screen-Coulomb interaction. For Byexitons, because you're looking at 4-particle excitations, the approach, in fact, involved 36 diagrams of this form. So this is one of this diagram plus 35 other ones. With the help of some really, outstanding collaborators, we were now able to do this kind of calculation. For example, we have applied this approach of looking at 4-particle excitation to understand and discover a new mechanism for singlet fission in molecular crystal. This is a direct Coulomb mechanism which has been neglected before. And also we were able to understand the Byexiton in the photoluminescent experiment of the monolayer tungsten diselenide that I showed you earlier. So let me first let me focus on this Byexiton in monolayer tungsten diselenide. Tungsten diselenide monolayer is one of these 2D materials. It's a semiconductor. It's Brillouin zone for the experts. It's a hexagon, and the low energy excitations are at the corners of the Brillouin zone, the K and the K prime valley. This is the band structure, and the spin texture of the state at the 2 valley are related to each other by time reversal symmetry. So if you look at the 2 valleys, it turns out that because electron-hole interaction is so strong in 2 dimensions, you could form very strongly bind excitons with binding energy in the order of ½ volt for such 2D materials. You could have optically bright excitons in one valley, for example, this one given here, or you could have dark excitons within one valley because if this excitation involves a spin flip, so it's not optically active, or you could have an inter-valley dark exciton, which have a finite momentum, therefore it's not optically active. Now, when you excite this system with high intensity light, then you could create a lot of these excitations, and you were able to also create a bond pair of two excitons, and that's the Byexiton we're interested in. So here is some calculated results we obtained by solving the four-particle Green's function from ab initio. Let's first look at the trions. In fact, there are two species of trions. This is our calculated binding energy in millielectron volt. It compares very well with experiment. So these binding energies are 30-some millielectron volts. So this is actually room temperature phenomena. And for the Byeexitons, the prominent species has about binding energy 22 millielectron volts. So this, in fact, of the first fully ab initio Manybody, the n+4 particle calculation from, it gives a very rich energy and spin valley level structure. The results, in fact, give us a lot of information and illuminate the experimental observation and actually give some new predictions. For example, in the interpretation of the experiment, it has been believed that the Byexiton, in fact, is a pair of one optically excited, optically bright intra-valley exciton pair with another with a dark intra-valley exciton. But we find, in fact, this is not what's observed. What's observed, because of Manybody effect, in fact, is a Byexiton correspond to a binding of intra-bright exciton with an inter-dark exciton. And that gives you the right energy and also many of the observed features. Now let me turn to time-dependent and nonlinear optical phenomena. Here, as I mentioned, you have to use a non-equilibrium Green's function formalism on the so-called Kaldis contour for the experts. What's important to mention is that now you have to solve such equation with two time variable involving the self-energy, which depend in principle on all previous times. And then the observables, say like the polarization of the system as a function of time, can be now then obtained from the time-dependent Lesser Green's function. This kind of problem is very challenging because it involves the time evolution of two coupled time variables. So it's scale like t cube. We know that even linear scale like molecular dynamic is bad, so TQ is really bad. As a first step, we have make a adiabatic approximation in that that we could ignore the memory of the self-energy. Then you could transform this problem into a single time variable problem. This is equivalent to the TD, the adiabatic approximation in time-dependent density functional in a similar spirit. Even within this approximation, it captures all the self-energy effects within the GW approximation and excitonic effects. It's accurate in moderate field regime, pump field. If you do it within linear response, in fact, it's equivalent to solving the beta-salpeter equations. But if you do explicit time propagation, the time then you will get out the time-dependent as well as nonlinear responses with many electron or excitonic effects included. The important thing is that within this approximation, you could do single time propagation. So let me show you just one application of this. This is the looking at field-driven phenomena in angle resolved photoemission experiment. That is, suppose you pump the system with a short pulse, and then later you come in with and do the normal photoemission experiment. This pump field might drive the system into a different state, and you could see different phenomena. So here, I show you some results where you pump the system with some frequency at 2.5 electron volt, which correspond to this monolayer germanium sulfide close to the band gap of the system, with an initial pump, short pump of 5 femtoseconds, and then you probe the system, do a photoemission experiment with a much longer time interval after a short time delay. So here is the calculated spectral function that is equivalent to what you measure in photoemission experiment. So what you see is the occupied bands and then some of the unoccupied bands occupied with, this is a calculation in TD-GW in the adiabatic approximation, neglecting electron hole interaction. But if you do the full calculation, including the electron-electron interaction, then what you see is in fact something very different. That is, you're seeing extra features, and these extra features are in fact due to excitons, coherent exciton being excited and drive the system into a different system. And just by reading off this extra intensities, you could see, you could find that there are in fact 2 series of excitons, and the intensity as a function of K actually give you a description of the wave function of the exciton at in K space. And then finally, one could also see a band renormalization due to the pulse of light that you send in the beginning. So let me now, in the last couple of minutes, if I still have a couple of minutes, I talk about our strategy of moving forward this kind of study on H.P.C systems. In particular, the exascale system that the Department of Energy, DOE, in the US are building right now. Exascale for the DOE really means GPU accelerated systems. So maybe in this series of seminars, you have heard about some of the machines that will be coming up soon in the US, exascale machines. Summit is the current fastest machine in the US. And this is a pre-exascale machine at Oak Ridge. And then the exascale machine will be these 3 machines. Perimeter will arrive at Berkeley next year, Aurora in Argonne National Lab, and then Frontier later at the Oak Ridge National Lab. The important thing is that it's not so easy to port the codes to this kind of hybrid CPU, GPU systems. So our plan, in fact, is to try to get ourselves involved with the pre-delivery machines to make sure that our codes will be able to run efficiently in this kind of machines. So why is it so different? So here I just compare the features of a CPU and versus a GPU. Here's the Cray CPU that's being used at NERSC at Berkeley right now. And this is the CPU at Oak Ridge, GPU at Oak Ridge Summit Machine. So for the CPU, like the one from Cray, you have 68 cores, 4 threads each, and 8 Y vectors per thread. So that means you get about 2,000 parallel lanes. On the other hand, the GPU, like this one from NVIDIA, it has 80 streaming processors, 64 by 32 threads per processor, and that means they have 150,000 parallel lanes. Now, for supercomputers such as Summit, it has more than close to actually to 30K GPUs. So that means it requires a billion-fold parallelization going from a CPU machine to a GPU machine. And in fact, the GW, the Manybody perturbation theory approach, is actually primed for exascale because there are multiple layers or parallelism one could exploit. Is much better suited for exascale system than, for example, DFT type of calculation, because within this kind of approach, you have non-local and frequency dependent operators. So our transition to exascale is just beginning. Our roadmap is to try to combine the state-of-the-art applied math and algorithm improvement with the H.P.C best practices. So we are about to complete the porting of our Berkeley GW code to the GPUs, and we will be also implementing other optimization for higher level theory, such as the time-dependent GW I mentioned to you. And we're going to continue our engagement with experts from GPU vendors and DOE user facilities. So in the future, we go to try to make as much knowledge transfer or programming skill and best practices as possible to other scientists and users. We're going to try to facilitate user and computer scientists at the very center to gauge performance of our and other people's software, and then further expand the broader user community for this kind of exascale applications. So let me just show you one example of our recent experience at Summit, which is a hybrid CPU-GPU system. We have this example is for a calculation involving a die vacancy defect, which involves 2,700 atoms in a supercell with about 1,100 electrons. This is the scaling in terms of petaflops per second, petaflop as a functional number of GPU. There's about 30,000 GPUs in Summit. So you see that we could scale linearly all the way up to the full machine. Here are the details of the calculated calculations. So the bottom line is with this kind of approach and our code, we could now scale up to the full Summit machine at Oak Ridge, reaching nearly about 40% of the theoretical peak from performance of the machine. It's approaching about 0.1 exaflop. These are double precision calculations. For this very large system, looking at die vacancies, the time to solution is only about 7 minutes. Compared to the Cray CPU only machine at NERSC, the code actually runs 100 times faster and give you a 20 times saving in energy penetrate. Okay, so I think I've completely run out of time, and let me finish up by just acknowledging the work. The work that I talked to you about is basically funded by the US Department of Energy, the National Science Foundation, and of course, the support of the Center for Computational Science, Study of Excited State Phenomena in Energy Materials. And all the results I showed you are calculated from using the Berkeley GW package. And you could learn more about that on this website. And these are the people who are my colleagues at the center, C27. Okay, thank you for your attention.
Well, thank you very much, Stephen, for this thorough description of the science and on all the efforts that are done in the context of this center. I remind the people attending, in case you were late, that you can always pose your questions to Stephen's presentation or the subsequent ones through the YouTube channel or by emailing to director@ccam.org. And we now move on to the second speaker of this afternoon. So now it's your turn, Claudia. I think you are muted yourself. You have to mute.
So, oh, I think it should work.
Yes.
Yeah, thank you very much for the introduction. I'm really happy to be here and share my thoughts about how to combine computational material science, solid state theory, and artificial intelligence, and also big data, in order to detect novel things and to put physics and research on a new level, if you like. And all this, of course, needs quite some support by H.P.C, which can, I think, make it happen. So let me take you first to a little trip back in time. So just to remind you how our science came across. So we started with experimental science, not us, but our ancestors around 4,000 years ago. And only 400 years ago, the first laws came into play and setting up the second paradigm of science, of physics, with theoretical physics. Of course, everything very much further evaluated and evolving all the years. And still, I mean, it's incredible how the precision of experiments now is increasing, and also what we can do, as we've just seen nowadays with our theoretical tools. But it's only, let's say, 40 or a little bit more years ago then the third paradigm came into play, and this is computational science, or computational material science in particular, because the advance of computer power and now H.P.C helped us to create a lot of, let's say, new types of research, or a new type of research, which is enabled by H.P.C and allows us to do new types of experiments, and these are computer experiments. In that sense, it's different from just using the equations that we have here, but using the computer power to solve these equations. And all these three pillars here, these three things, they actually form the tools that we have in hand right now in order to carry out our research on our daily basis. And this is where I would like to take you right now. And question what type of physics we do nowadays, right? And with this, I would like to introduce my group to you, actually. I don't do it at the end, I do it here. So you see here, the group is pretty big, and they're all sitting at their computers. Well, some of them are doing paper and pen work, because we also develop methodology, not only computational tools, how to implement these computational tools, but others, they're using either their own PCs, or some of them, they're using computer clusters, or H.P.C facilities in order to do their work. Now, why I'm telling this? Because I would like to share with you my thoughts about whether doing physics this way is the only thing we can do, or we can do more. So at the end of the day, what the people do is they publish papers, right? So these are very nice papers investigating different things, different materials, different methodologies, and things like that. And I would like to take one of them to give you a little bit more details about that. It's not that I'm going to talk about the physics in there, but I like to give you, let's say, a minute wrap up of what we did in this paper. So this is a paper about thermoelectric materials, and what you see here is actually a class rate. Class rates are formed by something like silicon cages or materials alike, and these silicon cages, they are semiconducting. So this is something we really like for thermoelectrics. They should be semiconducting. However, what we also like to have in thermoelectrics is a low thermal conductivity. And this is not fulfilled with these pure silicon cages. So what our experimental friends are doing, they actually put this, what they call rattlers, inside the cages in order to lower the thermal conductivity. That's nice, but it also comes with a drawback, because barium and strontium are ionic species. So what they do is they dope these cages and make these cages metallic. So in order to get back to the semiconducting state, we have to replace some of the silicon atoms here by species, for instance, aluminum, right? And then at some aluminum concentration, we call this the charge compensated states. It's 16 aluminum atoms. In this case, the system should go back, according to Sintel's rules, to a semiconducting stage. Well, what you see here from this picture, and on the x-axis, you see this aluminum concentration here. And if you focus on this green dot here, then you see that by looking at the density of states that is plotting here, that indeed at these charge compensated states, the ground state is indeed semiconducting. That's fine. However, there are other configurations of where these aluminums could sit in this cage. And taking out one of them, this is the orange dot here, and looking at the corresponding density of states, then you see there's a finite density of states at the Fermi level. So this state is metallic. So bottom line, that this charge compensated state is good, could be a nice thermoelectric material, but it could also be that it's not operating so well at high temperatures, but because at this high temperatures, the system could become metallic. So I'm not going to discuss the physics, as I said, but I wanted to give you some insight of what we conclude from a paper, for instance. Now, I also want to mention, at least on passing, that in order to get to this charge compensated state and all these ground state structures, by just trying out where these aluminums would like to sit in the cage, this is a combinatorial problem that no supercomputer on Earth could solve for you. So even any exascale computer couldn't do that. So in that sense, what we're employing here is a cluster expansion, which somehow shortcuts the whole thing. And it's a regression problem. So I think nowadays people would call it a machine learning tool. At the time it was invented, nobody called it like this. But just this on passing, that already now supercomputers and H.P.C is one solution, but we also have to think about models and tools in order to shortcut things. So let me get back to the paper about the publication. So what do we find in a publication typically? A table, yes, we have this in this paper. Don't try to read it, it's too small. There's a figure. Well, we have a few figures even in this paper. A structure, well, you have seen the class rate. Maybe we also extract a few other numbers that we have learned on the way when creating this data. But at the end, whatever we publish, it never ever can be the total information that we have learned in the 1 or 2 or maybe several years we have performed all these calculations and looked into this material. So in this sense, we have created very much knowledge by H.P.C capacities, but we never pass on this knowledge to someone else. So at the end, we have to say that our daily research is great, but in view of the problems that we like to solve, and we have the potential to solve with our tools, this is a very slow process. And I don't want you to get me wrong, because I really love to do the research that I'm doing with my group on a daily basis. But I think we can do better. And this is what I like to discuss with you. So how to exploit all the H.P.C capacity that we have, and maybe also shortcut things by not repeating calculations, so avoid calculations. It also is a speed up. It's also an aspect of exascale, right? So let's dream, and let's dream that someone, whoever it is, gives us all the data from all calculations that have been performed so far. And this is an enormous potential, because there's an enormous amount of information there, which nowadays is typically thrown away. What could we do? And in order to give you an idea what I'm talking about, I really would like to exemplify this with one number. This is the thermoelectric figure of merit. So I'm getting back to this problem of thermoelectricity, but not in terms of physics, but in terms of data. So as we know, of course, thermoelectric devices, they have to operate on a very large temperature gradient, because we want to recover waste heat from any kind of machines where the temperature is increased because its energy is not used. So that means that whatever we compute, we have to compute it for a certain temperature range, a large temperature range. And this is the geometry of the material. This is phonon spectra, because we want to learn the thermal conductivity. This is electrical conductivity. And now, if you look at the spectra of methodology, then of course, geometry would get from density functional theory, and the phonons you would get from Manybody perturbation theory, or even better, the electrical conductivity you would get from Manybody perturbation theory, density functional perturbation theory, or better, Abonesian molecular dynamics would give you phonons and thermal conductivity final. So if we now look at the right side here, what this means in terms of data, then you realize that just for getting out one number at the end, because thermoelectric figure of merit is basically the ratio between electrical and thermal conductivity. So for getting out one number, we have created not only gigabytes, but probably terabytes of data just for one material and one temperature. And throwing all this away is a huge waste of computational sources and also human sources, because there's knowledge inside. So, and this was actually the reason why we started with the Nomad repository. So this is a database of computational materials data. At the moment, it hosts already more than 100 million calculations. And we invite people all over the world to upload data from wherever they come from. And this, of course, makes data, so we're getting organized, that's fine, but it makes data also findable. And findable is the SF in the FAIR principles. And just to remind you, so we made this data repository public in 2014. This was 2 years before the FAIR principles were formally, let's say, written up or wrapped up. So we made or made a data infrastructure, a FAIR data infrastructure already public 2 years before that. Now, F is the findable. What about accessibility, which would be the A in FAIR? And of course, if data are uploaded, you can download it. So data are typically accessible because they're now sitting somewhere in the big database, in the cloud. No, in fact, actually, it's not sitting in the cloud. It's sitting at the Max Planck data facility in Garching, but it's not a big problem. So is this data accessible? And I would say, yes, it's accessible for the experts, because these experts, they can use the input files. They can see whether they get the same results. They can start looking at something similarly or slightly different without repeating calculations. So it's very useful for those people. So useful also for my group and probably for yours. But it's not really accessible to engineers who want to know what is the outcome of such calculations. And that's why we have created the Nomad encyclopedia, which makes data human accessible. So you can search for a material according to the certain criteria, and then the database or the graphical user interface would show you what the data are, probably what they mean, also giving you some description of this. So this is the A, but let's recall that these 100 million calculations, they come from very different sources. So from many people all over the world, but also they have been created by many codes, right? And so at the moment, we count approximately 40 different codes that is used by the community. And that means they come in their very different formats, very different units and whatnot. So in other words, what we had to do is we had to write parsers, one parser for each of these codes. And this parser, they also work like converters to the same file formats and units. And the prerequisite for this, and this is crucially important, that's why I make here as many exclamation marks as I can do, you need a full description of the data. So metadata are crucially important. Otherwise, we would never know why the total energy zero of WASP is something different, like in Abinet or whatever code you like to. So interoperability, in fact, is a very big issue. So even if you provide now normalized data, this means unified formats and units, this is simply not enough. So this is only the very first step. And I will get back a little bit later to this because I think interoperability is really the biggest challenge that we face in terms of fair data. Now, let's dream further. So we not only have theory, so we were talking before about the 3 pillars of material science. Of course, we also have experimental data around, and we would also use all the information about the synthesis of a sample, because if you don't know how the sample has been created and what the composition is, also the experimental data wouldn't be meaningful. But anyway, let's assume that all this is solved, so interoperability is not an issue for the moment. Then we can dream further and imagine that someone brings all this data together. So, and then we can fulfill actually or go in the direction of our scientific vision. Now, what is the vision that we have in mind? We would like to tell you, an engineer, whoever, where to look at if you want to find new superconductors or new transparent metals or new solar cells or thermoelectrics or thermal barriers or whatnot. So how do we get there? So in other words, we would like to draw maps where it tells us where to look at if you want to find materials for one application or the other. This is like you create a map of Europe, like, so you're looking at the map, you will immediately see where people may speak Spanish or speak English or speak German. Of course, the crucial thing is here, what is behind these trends, such that we are able to group them and to find these patterns. And here, of course, the descriptors, so these parameters that describe the property or the function of a material best, are crucially important. So this is what we really have to do. We have to find these descriptors that tell us how to tell different ranges of materials apart from each other. So, and let's get back to this. So all data, synthesis, experiment, theory. Now we can do everything, right? And still, I'm asking the question, is this enough? And unfortunately, the answer is actually no. And why is it so? And this gets me back to our time travel. So what we like to do in the future, and this is getting me back to, let's say, our vision, our dream, is to find patterns and trends in materials. And I think this will be somehow a very strong focus and pillar of our future research. Not exclusively, of course, all this, what we have right now, will stay in place, but this, what we call the fourth paradigm of science, big data-driven science, will actually be a very, very strong component of our future research. So for this, I think it's not enough to have our laws, and it's not enough to have our very nice computational tools, but we need more. We need artificial intelligence tools in order to find these trends and these patterns. Now, the first paradigm, of course, is something that is not unique to material science. It was phrased in 2007, the first time by Jim Gray, but of course, it's also very true for material science. The question is now, why did I say no before in the answer of whether having enough data is enough? Why is this so? Because you could argue that we have many areas that develop tools. So why can't we just use them from the shelf? So of course, there's computer science developing tools. There's cheminformatics, which is actually a pretty old field, much ahead of physics. There's signal processing. There is social media, not to forget. I think probably social media are among the biggest drivers of artificial intelligence tools right now. So in that sense, you can find many, many, many procedures, many tools that you can just use. And what I like to emphasize here is, yes, we can use them, but we can't use them from the shelf, because physics is very intricate. So all the interactions that we have in a material that somehow govern a function is much too complicated to learn without the input from the domain. So domain-specific knowledge is extremely important here. So let me get back to our dreams and to our maps that we would like to create, and emphasize a little bit what we need for our research of tomorrow. So we definitely want to exploit these trends and patterns in order to draw these maps. That's right. We will use artificial intelligence for this, but I like to emphasize that we indeed need to create novel tools. And novel tools means that very important to go beyond interpolation, because many of these machine learning tools nowadays, they are perfect in interpolating data. So it's like a fit. You increase the number of parameters and you get a perfect fit. But it doesn't tell you whether these methods really have power, predictive power. So in that sense, I think we're just at the beginning of developing tools and methods that allow us to have more predictive power and go beyond interpolation. So that's one thing. The other thing is just increasing the number of data is probably not important or not good enough, because we also need the relevant data, right? So we don't want to have more data on, don't know, of the water, and we don't want to have more knowledge about the sand underneath the water. We really would like to have knowledge on the pearl. So we would like to find the outliers, so those things, those materials, those functions, which are really important and are relevant and different. So novel tools and relevant data are definitely very important ingredients for our research of tomorrow. And just to give you some idea, if you want to look it up, there is this Nomad Analytics Toolkits that already shows some of these materials maps. So one of them, for instance, telling apart binary materials that crystallize in one or the other crystal structure. It can tell apart metals from insulators, or it can distinguish between topological and trivial insulators. So you can all this look up in the analytics toolkit and even play with this data. I don't have time right now to go into details, but I think it's fun to play with this. So let me get back now to our visions and also to our time travel, and let's get in the future. So where are we on our way to realizing what our vision is? For this, I get back just to remind you that this Nomad laboratory, this was established by Matthias Scheffler and myself. It really, let's say, came into play through a collaborative project on thermoelectric materials, where we realized how important it is not to throw away the data, but to use them for being reused actually. And this became a much bigger enterprise in 2015, so that we had the first round of centers of excellence, European centers of excellence. So at that time, we had a consortium consisting of 8 scientific groups with very complementary expertise and 4 high-performance computing centers to help us set all this up. Right? Where we are going now, and in order to discuss this, I'd like to get back to the question that I was asking before: what really impacts the eye, the interoperability? And this has to do with quite a few aspects or parameters. First of all, there's a huge variety of methodology in theory, in experiment, in synthesis. And this comes with a lot of veracity, of course. So you need to ask the question whether you really can compare data from different sources, even from the same experiment coming from different samples or different apparatus. It has to do with data quality. It has to do with trust levels. So we need to know which data to trust and to which extent we can trust it for which purpose. So we also need benchmark data in all respects. We also have to keep an eye on hidden parameters, because there's many things that are never written up, or many things that are probably not even obvious, because in theory there are computational tools that always come with some specific implementations and algorithms. So keep an eye on this, and all this somehow not only has to do with the interoperability, but of course also adds onto the problem of reproducibility of data. So why I'm saying this? Because what we are planning next, our next steps, they are taking care of these kind of issues. And one of this is, let's say, data quality, also variety, veracity, reproducibility, and trust levels. The other is how to go along, including also experimental data. So let me stay with the first one. And this brings me to the new round of European Centers of Excellence. So we are going to start actually in fall. The start has been postponed due to Corona, with NOMAD, with another NOMAD Center of Excellence. And I've written up here just a sentence that I would like to read to you. This is the essence of the project, right? So the NOMAD COE will assess and exploit the characteristics of next generation H.P.C for ab initio computational material science to enable investigations of systems of higher complexity in space and time, consideration of metastable states and temperature, and all this at significantly higher accuracy and precision than what is possible today. So what we want to exploit H.P.C capacity for is to putting theory on the next level. So not only, let's say, allowing for enabling more calculations of the same kind, but enabling better accuracy of the calculations. So what we want to use is the most modern tools and the most demanding methodology with highest accuracy, and bring these methods to the exascale. So in other words, if we look now at the spectrum of methodology that we have in hand, so for density functional theory, we can probably treat systems of a few thousand atoms in the unit cell. If we go to higher methodology like Green's function methods or even coupled cluster, there's no way of doing this. And so we need to actually push up these methods to make them available also for complex materials and systems. And this is, let's say, the plan that we have. So we have this pillar on exascale codes. Of course, we also need the corresponding workflows that help us to keep track of what we are computing. And of course, we also need to bring the data analytics to the exascale. And all this will then be demonstrated by use cases from energy and environmental science. So that's the plan for the NOMAD COE of the next generation. These are the partners. If you like to see more the people. So this is a very enthusiastic team contributing to this. We are looking forward to getting started. But at the end, I also would like to mention, as I already pointed out, that we're not only going to look in more detail and to establish better methodology in theory, but we also would like to include other aspects like experimental science. So, and that's why we have founded the FAIR Data Infrastructure for Physics, Chemistry, Material Science, and Astronomy. This is an association where we have brought together people from different areas in order to make the next step and provide a data infrastructure for functional materials, for applications of relevance for society. So, and this brings me basically to the end. I think if we build such a data infrastructure, FAIR infrastructure, that capture all aspects of data, and here I really emphasize that it should be synthesis, experiment, and theory, as well as data, metadata, and tools in order to handle all this, and novel concepts of artificial intelligence. So if we only interweave all these different components, then I think we really could go for this fourth pillar of material science and put the science, so to say, on a new and additional level. With this, I like to nearly stop, so to say. We already have taken a first step on this route. So this is a new project which is called FAIRMAT. So today I've been talking mainly about theory. We also will have an umbrella now. So like FAIR-DI is an umbrella for the whole thing. Now we have an umbrella for theory. We have one for synthesis. We have one for functional materials that we call use cases. And we have an umbrella for experiment. And since I think a few days or so, we already have the first experimental data, of course, in a very preliminary form, but we have the first experimental data already in the NOMAD database. With this, I would like to end. I hope that I could show you that somehow we have taken off in our way to our vision. Of course, we need to fly much higher, but the first steps have been taken. And with this, I would like to thank you for your attention.
Okay, thank you very much, Claudia, for this thorough presentation. And we will move now to the third presentation, through the third invite speaker. First of all, I would like again to remind you that you can always pose your questions to any of the lecturers today through this channel that you have in YouTube, or by email as indicated in the channel to director@cicam.org. So, as I said, we move now to the final speaker of today. It is Modesto Orozco. So Modesto, please, now it's your turn.
Okay, can you see my presentation, Ignacio?
Yes, it's okay. You can go on. Yeah.
So thanks, and thanks for the invitation. It's always a pleasure to help and to collaborate with CICAM. We have had two wonderful talks on material science. So I think my duties now are to talk about biology, and in particular, what we are doing, trying to join H.P.C and biology. This is a kind of disclaimer. So we'll summarize in this slide all my knowledge on computers. So I know this is a computer. I know this is not a computer, and that's it. This is all that I know. So, so I'm sorry if I say stupid things in the technical part of this presentation. Even I don't know much about computer. I was director of the Supercomputer Center in Barcelona, of the Life Science Department of the Supercomputer Center in Barcelona for more than a decade, and I learned a few things. I mean, when I arrived there, it was clear that H.P.C was in a kind of change of paradigm, in the sense that computers were not anymore based on very sophisticated high-end processors. They were commodities. They were made with normal processors, just putting one on top of the other. And it was also clear, especially when I leave, that Moore's law was not there forever. So this was more or less my period there. This is the power of the 500th fastest computer in the world. This is the aggregated power. And you can see already here that it was a decline, a deviation for Moore's law, and it's now very clear. It is now very clear that Moore's law is not going to work forever, that we are going to reach a kind of plateau in terms of the power of computers. And it's also very clear that you still gain power by adding more and more and more cores. So these are the 5th fastest computer in the world right now. In average, they have around close to 7 million cores. And every core is not extremely powerful. I would say a lot of power when you combine all the cores, but individual cores are not so extremely powerful. So we have a very specific tool. And I have been in all H.P.C committees for many years. And I have seen like two scenarios, two groups of people. So the H.P.C people is highly concerned about the users. And the users, so us, are very concerned about the tool, about the computer. And really, all of us forget the important here, that is the problem. What really should focus our effort is the problem, the type of problem that we have. And if you look in a historical perspective, the tool has, the problem, has guided the development of the tool. So we had a problem and then we designed a tool to solve this problem. So if you wish to put a nail, then you design a hammer. If it's a screw, you design and use a screwdriver. But this is not the situation right now in computer science. Computers, we don't control the way in which computers are. So, and then if we go with a particular problem, let's say that they have these balls, these screws, they will recommend me a hammer. And if I complain and say, okay, look, I don't need a hammer, then they will offer you a variety of hammers. And this is a real problem. And the real problem is that we don't control the tool. The tool is not based, as it was 2 or 3 decades ago, based on our needs, a community of computation, of people using computers. The tool is made based on other considerations. So the only thing that we can do, and the thing that we must do, is try to join and to understand in which way we can better use a tool that was not specifically designed to our problems. And then I put here side by side what are the nature of high-performance computing today in 2020, and what are the needs of the problems that are tackled in biology. So H.P.C is great for CPU-intensive calculations. The data doesn't used to be a major concern. The world clock is not really an issue. You send the calculation and it can stay there in the batch queue for weeks. Analysis is typically done a posteriori, not during the calculation. And in most cases, we're talking about a single calculation. So you have an input and you have an output. What is the type of problems in biology? They are CPU intensive, but typically not a single calculation. It's a composite. So you have to do many calculations. As a whole, they are very expensive. Data is a major problem. The analysis is typically integrated, is embedded in the calculation. So you run one calculation or the other based on the analysis that you do in the middle. The world clock is crucial, especially when you work in personalized medicine problems. You need to have the answer very quickly. And almost in all the cases, we are talking about multiple calculations. So you have multiple inputs, you have multiple runs, and multiple outputs. So problems and tools are very different. What can we do? We can, or we should try to build bridges. And this is what Byexiton, another center of excellence, the previous one, Claudia explained us, is to make these bridges. It's trying to join the tools that are there, that are supercomputers, with the problems that the biologists have. And I'm going to talk about a very small fraction of the things that Byexiton is doing. And particularly, I'm going to tell you about Byexiton building blocks. That is an initiative to actually try to close approach biology and H.P.C. I'm going to talk about something that I'm not an expert, to be honest, and is a work that has been done for many years now by a big team. I think that is a very nice collaboration between people in my group, IRB Barcelona, people in the GW and Rosa Badia at the Barcelona Supercomputer Center. I think it's a good example of how you can do things of real impact in biology from using the knowledge of computational biology. It's not to say Adam, that I think is connected, is actually the main person, you know, the main responsible having all this running. So what is the idea of the Byexiton building blocks? Is to build a software library. So typically a biological project implies to run many different jobs. Some of them are very quick, others are very slow, and you need to put together. And typically the way in which you put together depends on results in the middle. So it's a very dynamic. It's not always the same. It's not a standard, I would say, Gaussian job that you have an input and you run and you have the output. This is a very flexible, and then you need a flexible structure. And this is what we did. So we break the calculation into small pieces, and then we organize workflows. And then on top of that, we put a workflow manager. So a software that allows us to integrate these workflows and adapt these workflows to specific computers. And you will see that we did it with H.P.C in mind. This is a kind of summary of what I'm talking about. So we have different input files, we have different configuration files. Then we do a kind of input parameter adaptation. Then we have the layer execution that all these inputs are introduced, and then they got outputs, and then all these outputs are adapted to do further jobs or to get the final output. On top of that, you have a layer, a manager layer that actually make all this to work in an efficient way. Our BBB are fair. Claudia already talked about fair. Is one of the keywords in science right now. Of course, it's interoperable by definition. It's findable. You can find in many different repositories. It's actually accessible, from many different methods and models, and it's reusable. So it follows all the Jupyter notebooks, Dockers, and Condas, and all these interfaces. We have not one, we have many modules of BBB, of Byexiton's building blocks. Some of them which are like the basic infrastructure, the wires of the system. And then we have many that actually are mostly right now related to the field of simulation, that is the strongest field in Byexiton. When you go to one of these modules, then you see many individual building blocks that you have here, and then you can combine it. And as they are in Conda, you don't need to be worried about dependencies, about the version that you have. As you can see, we are not incorporated only Byexiton technology like Romax. We have also competitor technologies like Amber. And all this is packed in a way that you don't need to worry about where you're going to run the simulation or what is the type of computer where you want to run the simulation. Let me just put one slide about the syntax to see how simple everything is. For example, I'm going to talk about this specific building block, but this is how simple it is. So that you have an input file that is a PDB, for example, the PDB of this protein, and then you want to generate many mutations of this protein, and then you just put the list here. For example, here we are mutating this arginine here into an alanine. It's so simple. So then you will do all the mutations, and of course, on top of that, you can run simulations on all these mutations. I will show you in a second, but let me finish this kind of technical part, showing the effort that we have done to put this workflow philosophy that is typical from bioinformatics into the field of H.P.C. Mainly when you talk about H.P.C, you talk about scalability. You want to use all the processors that you have annotated. So you have, in this particular case, we have used actually more than 50,000. Examples are for 38,000 cores. And I'll show you that we use 100% of them in a sustained way. And this has been possible thanks to a very nice collaboration with the BSc that have developed this wonderful interface named PyComs. So how we modify these BBBs and these workflows to work in H.P.C, very little changes. We divide into files. This is a Python script that explains what you want to do, you know, the loops, everything you want to do, and then you have a down, you have the parameters, so the inputs, what are the, you know, all the characteristics of the simulation. I'll give you an example because this is just a part of the Python file. Then it'll give you, you know, what you want to do here. You want to do mutation, then you want to run free energy perturbation on this. And then this is the data. For example, I want to do this mutation. So arginine 5 to alanine, arginine 5 to glycine, arginine 5 to lysine. And I want to do this many times and then generate the structure, do the dynamics, the free energy perturbation, etc. So very, very, very simple. How we managed to run this in a supercomputer, this is PyComs, this is the work of the group of Rosa Badia. It's just very simple. So as you see here, for the Python is a simple loop, and then PyComs recognize that this loop are individual calculations, and then put all the dependencies on the flow chart, and then decide where to send what. And then use in a, you see that in some cases there are different routes, in other cases data need to be joined together, then PyComs take care of everything, and then use all the processes that you have accessible. I show you here a few examples. Oh, I think only one example. This is of the results that I'm going to show you in a while. And this is on the calculation on 2300 nodes course in Mare Nostrum. All these workflows are running in different tier 0 machines in Europe. But what is interesting here is you see that for as long as we have, of course, input, output and input are always a problem. But for most of the time we are using 100% of the machine, so full efficiency. And I'm going to give you an example. All this was technology. I'm not very proficient in technology. I'm going to now put an example in my area of expertise, that is computational biology. And this is an example of a pre-exascale BBB workflow centered in COVID. And this is mostly the work of Adam again, and two postdocs in the group, Vito and Miłosz. And you have been probably in an Avogadro number of COVID talks. I have been in many already. So I'm not going to bother you with details that you probably know. This is the guy. This is COVID-19, SARS-CoV-2. And this is the famous spike protein. This is the protein that is actually responsible for the virus to be recognized by the cell. And actually, this small part here that is called RBD, this domain here that makes the interactions. So this is a spike, this is 1, 2, and this is the receptor that is called angiotensin II receptor. So it's a protein that is in our membrane and has nothing to do with that. It's a protein that is involved in the control of the blood pressure. So not any reason why this is recognized by a virus. So the point is that this complex is formed. When this complex is formed, many things happen. I'm not going to go into the details, but at the end of the day, the capsid is on top of the membrane and is incorporated, and then, Houston, you have a problem. Already in 2019, when the first paper was published, it was clear that the RBD subunit, so this subunit here that is recognized by AC2, was similar actually to naked eye to pangolin. Pangolin is a very exotic animal, a very exotic mammal from China. So the RBD of pangolin and the human virus SARS-CoV-2 was very similar. But on the contrary, when you consider the entire spike protein, it was very clear that the closest one was SARS-CoV-2. Of SARS-CoV-2 was rhinolupus, so was RAG13. What was a virus that was infected rhinolupus, that is a finis, that was a kind of bat. why a virus that was infecting bat take a portion that was similar to pangolin virus, and actually infect human is something that nobody can understand. But we can actually take a look at what happened. And for the bio here, I'm sure this will be shocking. What you have in the left is the number of mutations that happens between the virus from bat, rat G13, and SARS-CoV-2, so the virus from human. And you see that in a very small domain, we have more than 20 mutations, and many of them are dramatic mutations. So you are modifying the charge, things that you will never see by plain mutation. And on the right hand, you have the differences between the human and the bat AC2. So the receptors are quite different. So we are not very close to bats. So our angiotensin II receptor is quite different in this region. So how we move from here to here to adapt the changes from here to here is not clear, but I will show you a little bit what we learned. So what you have, and these are results from Vito. What you have here is the AC2, so the receptor from the bat, from Affinis, the receptor from human, and you can see that there is already a quite different change. So you see a lot of red here, you see a kind of gorge of negative charge here that is not present here. I will show you in a different way. This is the complex that was known by electron microscopy, and this is the case. This is AC2 from the bat. This is rat G13, so this is the bat, and the virus for the bat. And this is human protein, and this is the protein from SARS-CoV-2. So our virus. And you see that there is a dramatic reorganization of the electrostatic potential. Okay, then the question is how this virus became infective to human, and whether this virus is going to be infected to bat. And this is a type of H.P.C problem. And here I don't have time to go to the details. This is a type of calculation that is called potential of mean force. And in short, what you use is statistical mechanics, full atomistic description, and we use a variety of the technique that is named metadynamics. So at the end of the day, what we do is to take receptors that were far away, and then we approach each other and compute what was a binding free energy, what was the chain in affinity. And these numbers are, you can't trust these numbers. These are still on process. So these results are not converged. So this number may be here, but there are a few things that are clear cut. These are not going to change. And these are, I think, very surprising. The first one is that the human virus is ultra infective for humans. So you change from here to here. There is a dramatic gain in stability when these two guys approach each other. If you try to infect humans with the bat virus, this is not easy at all. It is favorable, but it's a very low free energy gain. And the surprise, and the second surprise I would say, is that actually the human virus seems to be quite effective to bat. And this is maybe a problem in the future if we think in zoonotic expansion. So what our calculation, these are calculations for MIWASH, are actually at this point suggesting is actually that the change from rat G13 to COVID, SARS-CoV-2, was not a change to gain infectivity for humans, was a change to gain general infectivity. This guy is a professional infector. By changing all this electrostatic pattern, it managed to get a very unspecific, if you want, but very high affinity way to infect other organisms. Okay, so we can do this for a couple of viruses, a couple of receptors. We cannot do this for all the variants because we are again in a combinatorial explosion. So we have many viruses to study. We have many potential receptors, we have many variations of SARS-CoV-2, we have many polymorphisms of the human AC2. We need to do other things. And this is where our workflows that I summarized before came in action. It was, again, a collaboration within Byexiton with a group of Verdegrot in the Max Planck that they built this wonderful PMX. I don't have time to talk about PMX. It's a kind of free energy calculation. It's a non-equilibrium free energy calculation that is actually based on running many non-equilibrium simulations and then using a Crook theorem you integrate to get a reversible number. So you can see here reversible works, you integrate reversible works, and you get irreversible works, and then you have reversible works of free energy. So to make this in the high throughput is nearly impossible, but we managed to do it thanks to this workflow. So the idea is that you take a structure, you generate mutants, you equilibrate, you run the free energy simulations, and this is an example. Again, these numbers are not converged because we are talking here about 23 mutations. It's a pain on the neck from a technical point, but we are getting something between 7 and 15. So this is actually confirming that RAG13 is not great infecting us, and actually this movement was a dramatic change in infectivity. And as I show you here, this gain is actually general. It's not typical, it's not only for human. So when you run the simulations from a finished to human, you don't see much changes in infectivity. So RAG13 was not a great infector agent for bat. And COP2 is a great infector for bat, human, and probably many other organisms. So let me see how far can we go with these workflows, integrating pieces in the workflows that came from bioinformatics. And this is a tool we developed time ago, again with the BSc, with Visual P. It's called PMOOD. And this is a typical bioinformatic tool, but was designed from the beginning to run in a H.P.C center. So what we can do here, and this is a machine learning, what it has inside is a random forest. And we mine all the data that is already available on mutations. And as we run in an H.P.C center, we are able to do all the possible mutations, not as many others to explore the impact of a single point mutation in AC2, in the receptor, but to explore all the possible mutations. So what you have here is a position, for example, position 200, and then you have the 200 modifications that you can have. The 20 modifications you can have in the 200 position. And then we do for all the protein. And okay, what you see here are regions that are in red. Regions that are in red are regions that are very tightly preserved by evolution, because if you mutate this position, this means pathology. And you don't want to have pathology in a protein, pathology related to a protein that is in control of your blood pressure. You don't want to, your heart to explode because you are not controlling blood pressure. So you see a lot of red here are regions where you can't expect massive changes, massive polymorphisms. And you see other regions that are in green. But then you can plot on top of that those polymorphisms that are in human, those polymorphisms that appears in human population. You have here the polymorphisms, then you have here the type of change that you have, the amino acidic change, and you have the allele frequency. So as SARS-CoV-2 is recognizing this protein, you can expect that these mutations may affect COVID binding. So, but where these mutations happen, I'm sorry, I don't have the slide. Sorry, I missed the slide. But this, I should tell you, these mutations happen always in the green region. So all these polymorphisms that you have here that can be related to COVID are actually centered in places which are not functionally important for the protein. So the virus has managed to get regions to interact which are not ultra conserved by evolution, regions that are mild for the function of the protein. But we can then adapt our PMX BB workflows to study the impact of human polymorphisms in the binding of the virus. And again, these are work in progress, but few things are clear already. Remember, what you have in green are the expectation of how should be, in terms of the protein functionality, what should be the impact. Most is green, so not big impact. And the numbers you have here, some of them are still running. The numbers you have here is the impact in binding of the COVID. What can you learn? What you can learn is that in general, there are few polymorphisms that are gonna make you more infective for the virus. There is this one that actually will make you more susceptible to the viral infection. Not dramatic, but it's like a 10% more susceptible to the viral infection. And there are a few of them which make you, I would say, fully resistant to the viral infection. Of course, this is just, we don't have experimental validation. In principle, if you have this polymorphism, our simulation suggests that you should be free of the virus. The only problem is that it happens in only 1 of every 100,000 or 4 of every 100,000 people. So are not very common polymorphisms. Where are we going now? I mean, we are going in trying to say where the virus is going to go. So we can repeat this with our workflows, not for a single mutation. I showed you before, we can study many, many mutations. We can move from one receptor for one species to another receptor for another species. And we'll try to use this to determine what are the zoonotic expansion. And what really interests the human, what may be the final reservoir for this virus, where this virus is going to stay. If the virus stay in a very exotic species okay, this may not be a dramatic problem, but if the virus find a wonderful reservoir in the chicken, then Houston, we have a problem because the virus is going to be there for a long period of time. We can do these PMX-VB workflows adapting to another thing. We can study what is the impact of the virus variation. We have now thousands and thousands of viruses that have been sequenced, and we mine all this information and then take a look at mutations that have been in virus along the time, and see how frequent they are. So if you have one mutation that you find only once, that means that it will be like a random mutations. But if you find some mutation that stay there for many, many long periods of time, for a long period of time, it means that this mutation has been installed in the virus, so that the virus mutate by random to this specific, for example, 705 asparagine to lysine. This mutation happened randomly, but then it turns that this virus became very effective, and then it has a lot of virus based on these mutations, and then this mutation is stabilized and put more mutations in there. And what you have here is the result of our workflow with PMX. And this is a variant that is also in the workflow, that this is a data mining drive strategy developed by Luis Serrano at the CRG. It's called FoldX. This is a very fast strategy. It's a statistical base, again, based on machine learning approaches. You can see if you compare our numbers with their numbers, of course, there are some discrepancies, but in many cases, you see that they are not too different. Anyway, take a look at this number. So we are running this simulation. There are some that are we need to run it. But what you find is that when you have something that is very bad, this is a mutation that has no future. When you see something that is very good in terms of binding, is a mutation that is stabilized. And this indicates that this is a way in which we can detect mutations that are accumulating and are making the virus more and more effective. And this is something that is right now happening. But we can go to the future, and this is a collaboration with computational genomics in the IRB, Nuria Lopez-Vigas. She has built a kind of database of mutations that are possible. So these 2,200 mutations are not happening to the virus yet, but are mutations that are possible based on basic rules of genomics. And then using these workflows, we can actually analyze what is the future of the virus, what are the mutations that can happen. And for example, here you see, and these are good news, is that most mutations that may happen are actually bad in terms of infectivity, but you see a few of them which are, I'm sorry, better look at here. You see a few of them which are actually expected to be infective. And then what we are doing now is to take those mutations that has the worst possible profile and then move into the PMX and to have a more accurate number, a more accurate estimate of what is the impact, or what can we expect, what new surprises can we expect from SARS-CoV-2. And this was my last slide, and actually this is just to remind me to thank all the people that has been involved in this and many other projects. In the computational side, from my group, I will quote Adam here in the left, and user P, also from BSc, that are responsible of all the, all the hardwiring, all the development of these tools that make possible actually to use. If we have 100,000 core, we'll use 100,000 core to a problem that needs urgent computation. And thanks all of you for your attention.
Thank you, Modesto, for this very interesting talk. So now it's time for the Q&A session, which I'll be happy to start in a few minutes. Just an observation, we are running a bit late, I would say quite late actually. So I will ask questions in the order in which they have come in, but maybe I will not ask all these questions, and then I would ask the speakers if they are available to us emailing them the questions and trying to give an answer to those either on the SICAM website or via direct email in case in which we have received them via email. Is that okay for you?
Sure.
Excellent, thank you very much. So let's go back to the very first talk. And Stephen, there is what I suspect is a rather technical question, which is, do you also use KKR codes to simulate systems when, for example, they have impurities? As a non-expert, maybe I would ask you to spend a couple of words to explain what a KKR code is in the landscape that you have just described.
Well, I think the KKR code is a Green's function code that is at the 1 electron level. Usually that's very good for studying Fermi surfaces and so on. We do not use the KKR type of formalism. These are interacting many-body Green's function that we are focusing on.
Thank you. Then there is a question that came up during Claudia's talk, but I suspect that it could be interesting to ask it to all our speakers, although with different variations on the theme. And I mean, the preface to this question was the fact that there is quite a lot of excitement about the use of artificial intelligence and machine learning right now, in particular in materials. But do we actually have one example of a particularly important success story of something that has been achieved thanks to the use of AI and that could not have been achieved otherwise? Or are we still at a level in which we know that this is a promising method, but we don't have examples? And then maybe, Modesto, you could chip in and let us know what is the situation in bio. Claudia, would you like to have a first go at it?
I think, I mean, we have to distinguish here between two things. One is if you really want to have a very big progress, then, as I said, we need to develop new methodology, because just taking a method from the shelf, call it machine learning, and predict something probably doesn't take us where we want to go in general, because I think artificial intelligence has the power to put science on a new level, but we need, as I said, we need the relevant data for this, we need new tools and concepts for this. This is for the big goal. But then, of course, I think now, as I already pointed out in the beginning, everyone who does a regression problem calls it machine learning. So in that sense, machine learning has already solved many problems. So it's always a matter of perspective, right? But I really would like to emphasize the big goal. And there we are at the beginning. We have single examples already where it could go, where it could work. I've pointed out this artificial intelligence toolkit where you see some of these examples. But of course, we need to fly much higher, as I said. We need to go on in this way. Yeah.
I think that in the biology field is no question, on the power of machine learning approaches, and probably the best example is this competition called CASP, that is a competition where all the groups doing protein modeling join together, and then they make predictions. And there are groups that always do very well, and then there are like schools, and you know, they fight each other, saying that the method is better. And the past year, the competition was won. And actually, all the others were destroyed by Google. So Google built a team, a big team, and with very deep neural networks, they defeat all the human experts. So of course, biology is different to material science in the sense that the biological problem in terms of chemistry is very simple. So God was a good biologist, but a bad chemist. I mean, the type of chemistry that happens in a living organism is simple, and that makes things complicated is the magnitude, the size of the systems. So it's a problem that fits well with machine learning approaches.
Thank you, Stephen. Do you have any comments on that particular point?
No.
Okay. So then we move to the next question, which was asked to Claudia, and that is, what do you think of the AIDA and materials cloud approach, and in particular, the focus on computational provenance in the landscape of what you have described today?
I think as there are many codes available, so I just mentioned we have 40 electronic structure codes. I think also all the databases that we have right now take very different approaches, right? So our approach was we allow people from the whole community to participate at the expense of that we have to take care of the parsers and the metadata and interoperability. So we request input and output files. So in that sense, every calculation is reproducible, and we can also judge about the quality if you like. We won't do this because every calculation has its certain purpose, and the people who have run a calculation, they know what they have done, right? So, but in principle, by having input and output files, we know everything about the provenance of a calculation. So it's just a different approach. I won't say one is better or worse. I think this is different. They're orthogonal, and I think it's good to have this let's say zoo of different approaches, because this is finally what the community needs, and they can choose what they're going to use.
And then second part of the question from the same viewer, do you use or develop ontologies in computational material science within the normal AI approach?
Yes. So, I mean, we started with defining metadata, but also to already combine them, so to say, to group them, to build a hierarchy of metadata. So in that sense, our metadata items are already connected by a first version of an ontology. Probably it's not a full ontology, but it's a first step, and we're definitely going further, because we also want to include experiment synthesis as you like, and probably then also connect to other ontologies that are developed somewhere else to put this together. But it's a hard task, so it sounds much simpler than in fact it is.
Yeah, I guess so. Okay, that's all we have got from our audience today. Ignacio, do you have questions or curiosities?
I mean, I had a curiosity. So somehow I think in the three presentations, to some extent, there has been this question about how to address exascale, and also what is relevant from a scientific point of view. I think that maybe Modesto put it in more clear way by saying that we have these generic computers or machines that are not necessarily built according to what is the scientific question or problem to handle. So there is a question of how we can better make use of it. And there was this examples of more high throughput towards a long or high sort of strong scalability. So again, this is more an open question for the three of you. In your expertise or in your area or the problems you're working with, how do you envision really benefiting from this exascale? Which type of approach do you think it's more or offers you more possibilities of taking advantage of this tool? Say, there is also the question of this combinatorial explosion, that then again, it's more on the data, but then also how exploit that in an intelligent way. I don't know if you have comments about that.
I think I could make a comment on this. I think when you looking at many-body interactions and looking at the higher level theories to try to describe complex phenomena, then things scales pretty dramatically, like n to the 6th power or exponentially. So as long as you write your codes to take advantage of exascale, then having this kind of powerful machine will actually dramatically allow you to look at much more complex processes that occurs in interesting systems, because this is the scaling with the number of particles that's involved.
Mm-hmm. Okay.
I think that in the bio area, there is a point where actually exascale will be crucial, is to actually increase the accuracy of our models. So we're using potential functions that are really poor, and we know are poor. So as computers became more and more powerful, it would be possible to actually move to better description of the potential. The second area is actually that for most bio problems, you have a double problem. One problem is the calculation and the other is the data. And so we need to have the data close to the calculation. So we cannot download, teras and teras of data to run a calculation. So this is why an H.P.C center is fantastic, because then you have the data and then you have the computer required to manage the data in the same place. And this is something that with other solutions are not possible. And the other is the facing this type of problems. I try to give examples. Are problems that are a single biological problem, but implies many, many different calculations. So you cannot solve this by other ways. So you cannot run one calculation there, when, and then move from one computer to the other. You can do this with, in the workflow I show you, I mean, we study all the mutations just with one line in the Python code, saying do this mutation, this, and that's it. We need only one person, 10 minutes, and then the computer was used fully in this specific problem. These are type of calculations that I think are going to be of high impact in biology, and that's very difficult to be done out of the H.P.C centers. And of course, wonderful if this H.P.C center, they are now 0.5 exascale. I'm sure in the very close future will be exascale, and for biology will be very important.
For us, it's probably a combination of these two. One is for the approaches that are available right now, we can only do it for a certain system size. So if you really want to model a complex alloy, there's no way of doing this right now with even vanilla type of functionals. But if you want to do better, because we know that for many properties like be it barriers, band gap, whatever, you need to do more than a standard DFT calculation. So like what Stephen is pointing out, this will allow us now to use higher level methodology for more complex problems than we can do nowadays. And this will open also a new level of accuracy, I would say, because so far we very often are limited to take the functionals, the cheap functionals for complex problems, which is probably not appropriate enough for being quantitative.
So a curiosity, how transferable would some of the protocols that have been developed, for example, within the bio, to materials and vice versa? I mean, these tools to manage multiple realization of calculations instead of residual.
I would say that is embarrassing similar. I mean, some of the slides from Claudio may be my slides. So about FAIR, about metadata, about ontologies. So as everything is data related, I mean, many things are very similar.
Workflow
and I'm sure
management and things like that.
I think I show also the keyword workflow in one of Claudia's slides. So it changed the Hamiltonian, but yeah, I think many things we are facing the same problem, the same type of problems at the end.
And so then my next, maybe slightly naughty question, or maybe very naive question, is how much communication is there between these different communities on these specific problems?
I don't know. By training, I am a quantum chemist, so I'm a friend of many of the guys in the moment. but it's still, I think, from my point of view, it's still not, it can be improved.
But I think it will be improved and will be more intensified once we have made our first steps, because so far exascale technologies are somehow a white spot on the map, in particular because we don't even know much about how the next generation of computers will look like, and probably also those who build it don't know the details yet. So I think we have to explore first what really can be done, and I think once we have made the first steps, I think the interaction will of course increase.
Do you see a difference in the landscape in the US or Europe? Steve, I don't know if you are familiar with both.
I'm not familiar with both, but even within the US, DOE is talking to different vendors, and they have different architecture, and they use different GPUs. So I think from a point of view of the scientific community, we have to engage early with the computer builders, the vendors, to make sure that our codes and what we have in mind, that is the problem and the tool could match better. And that's what we try to do, because we're anticipating 3 new machines going to come out in the US, and we more or less know who are the companies that will be doing it. So there are selected groups that can be engaged with those developers earlier to make sure that scientific computations can be taken into account. The various codes that are in the community can be used as an input to the new machine that's being built.
So we got one more question, but in the interest of time, maybe I will forward that to the 2 material scientists to whom it is addressed. And I will now leave the floor to Ignacio for some conclusions.
I think it's, I agree that we are now running a bit late, so it's a pity to finish. But thank you to 3 speakers for these very illuminating presentations and this very nice and engaging discussion. And also thank you, all of you who have been following us during this webinar series. I hope this has provided a view on quite a number of initiatives around scientific interests and possibilities offered by the coming new environment in the H.P.C. And you can come back and watch these sessions. They are stored in the SICAM website and also in our channel, the SICAM channel in YouTube, so you can revisit and remember some of these discussions or past sessions if you haven't had the chance to follow them. And please stay tuned. We'll finish now. We will have a relax over the summer break, but we will be organizing new activities in the fall. We will advertise them, and you can always find the information also in our SICAM website. Thank you very much again, and I hope to see you soon back. Goodbye.
Thank you.
Thank you.
Bye.
Bye.
Ignacio, thank you very much for organizing this series. I enjoy it.
I hope. No, thank you very much. I think it was, thank you for waking up so early to join us. But yeah, it was a nice presentation, and I hope you enjoyed also the discussion. And yeah, thank you. Thank you. Yeah.
Thank you. Bye.
Bye. Bye. Bye. Okay. Oh, okay. That's funny. All right. Thank you very much. Bye. I don't know who is there, but bye. It's funny.